Hello friends, welcome to my channel Shamina Sarfadin and myself Shamina, Head and Associate Professor, Department of Biochemistry from Ratnaveli Subramaniam College of Arts and Science, Coimbatore, India. In this video, we are going to concentrate on what is antigen and antibody. To say more about it, specifically we are going to think what is an antigen and what is an antibody and what is the interaction between this antigen and antibody. And well, we are also going to research more about how the immunological response happens because of the antigen antibody reactions when an incoming pathogens affects the cell. So let's go in detail about it. So now let's see what are antigens. So when we hear this word antigen, something very much foreign to our body, something very new, okay? Those things, they are termed as antigen. Generally, how we can define it as antigen is a toxin or other foreign substances that which induces an immunological response. So these immunological response are going to be the fighter against the pathogenic infections. And an antigen, it is a substances that causes your immune system to produce antibodies. And when the antigenic substances is being very much capable to produce an immunological response, especially when they activate the cells such as lymphocytes, then we can say our body is strengthened enough to fight against the infections. And to say some examples of antigen, usually they are proteins in nature that are present on the surface of the cells. Example, viruses, fungi or bacteria. And the non-living substances such as toxins, chemicals, drugs and even foreign particles can also act as an antigen. So, in immunology, the antigen, generally we call them as a molecule that can be present outside a pathogen, which can be able to bond by a specific antigen antibody. We call it as antigen specific antibody, okay? Or simply we can also call it as B cell antigen. Those receptors, they are being termed as BCR. BCR means B cell antigenic receptor. And when there is a presence of antigens in the body is now triggering a lot. Do you know for what? Yes, it is for production of immunological response. And there is one more factor which we call it as TCR, that is T cell receptor. We all know T cells are very much good enough to come and fight the pathogens. And both this TCR and BCR, they are both highly variable antigen receptors. Okay, they altogether, they bring the immunological response. Generally speaking, antigen are targeted by antibodies, and each antibody is specifically produced by the immunological system to match an antigen after the cells in the immune system comes in contact with them. So the antigen is a molecule that binds to the antigen specific receptors. So the antigen may originate from within the body also. Sometimes they may be called a self-antigen. And sometimes they may not originate from us. They might originate from the external environment. At that time, how we need to name them? They are called as non-self. So generally, I can call them antigenous, self and non-self. So the immune system is supposed to identify and attack the non-self invaders from the outside world and thereby the harmful substances in the body, and usually that does not react to any self antigens under normal homeostatic condition due to the negative selection of T cells from the thymus. So now let's see what is an antigenic classification. Yeah, the antigen are being classified mainly into exogenous antigen, endogenous antigen, autoantigens, neoantigens, viral antigens, and tumor antigens. So look at the picture. Here is a bacterium, the bacterium or the viruses are considered by the body as antigen because they are foreign to us. Now let's look into detail about what is exogenous antigens. The exogenous antigens are antigens that which enter into the body by the following process. When you respire the air inside, that is when you inhale, okay? Along with your inhalation process, there are also lots of microbes may have a chance to enter into your body. So generally they used to say the process of inhalation, ingestion and injection are the process for the entry for exogenous antigen. And the immune system, they respond to these exogenous antigens. They are often subclinical in process by endocytosis or by the process of phagocytosis. These exogenous antigen that which enter into the cell are being identified by whom, there are cells called as APC, which we call it as antigenic presenting cell, and later they are being processed into various other fragments. And these APCs are present the fragments to the T cell helper cells, generally CD4 cells. So by the use of class 2 histocompatibility molecules that are present on their surface, just like that, the T cells are specific for the peptide pertaining to MHC complex. So now looking into the endogenous antigens. Endogenous antigens are generally within the normal cells as a result of normal cell metabolism, or simply we can say because of a viral or an intracellular bacterial infection. The fragments are being presented on the cell surface in the complex which are being restricted to MHC, that is major histocompatibility class 1 molecules, and often they go and activate the cytotoxic CD8 plus molecules which actually recognize them and make the T cell to secrete various toxins that may result in either lysis or a condition called apoptosis, which we call it as programmed cell death. And next, coming to the autoantigens, it is a very normal protein, or we can say it is a protein complex. Sometimes it might be nature of a DNA or an RNA. It can be recognized by the immune system of those patients they are being suffering from specific autoimmune diseases. Example, you can say rheumatoid arthritis. And under the normal conditions, these antigen, that is the autoantigen, they should not target of the immune system. But in case of the autoimmune disease, their T cells are not being deleted and simply they start to attack the pathogens. And now realizing about neoantigens. Neoantigens, they are the one that are entirely absent from the normal human genome as compared with the normal self-antigen. The neoantigen are in relevance to tumor control, which are the best quality of a T cell. that is being available for an antigen which does not get affected by the T cell tolerance. And next is viral antigens. These viral antigens, they are for virus-associated tumors such as cervical cancer, and it is often called as a subset of either head and neck cancers. And these epitopes, that is the epitope that are very much good enough to bind these receptors, they have been derived from the viral open-ending frames that have been contributed to the pool of neoantigens. And finally, to speak about tumor antigens. And these antigens are often the antigens that are being presented by either by the class 1 molecules or class 2 molecules of MHC. And these MHC class 1 and class 2, they are present on the surface of tumor cells. And these antigen, they found only on such cells, and thereby they are being termed as TSA, that is tumor specific antigen, and they result from a tumor specific mutation. So tumor antigens can appear on the surface of the tumor in the form of any form that can be sometimes mutated along with the receptor in which they can be recognized by B cells. Let's look into what are antibodies. An antibody is a protein. They are being produced by the body's immune system. You know why? They are encountering with the harmful substances such as antigen. And the example of those antigens might include the microorganisms such as bacteria, fungi, parasites, and viruses. And sometimes even chemicals may also act as pathogens. And each type of this of antibody that is being produced is very specific. They are unique in nature, and they are responsible for defending the body against the harmful pathogens that are enter into our body. And antibody being a protein, they are produced by the immunological response may recognize these harmful substances, antigen, and they act upon with it. So an antibody can also be called with another name, that is immunoglobulin. It is a large Y-shaped protein. When looking at the structure, it is just look like a Y-shaped. Say they are being produced by plasma cells that is used by the immunological system in order to neutralize, as we know, to digest or neutralize the pathogens such as pathogenic bacteria and the viruses. So the antigen recognize a unique molecule of pathogen, which is antigen in nature, via the fragment antigen binding site. The Y shape has a fragment which we call it as FAB. This FAB is a variable region. Each tip of the Y cell of an antibody, it will contain a paratope that is being specific for one particular epitope or an antigen, allowing the two structures to bind together and thereby bringing out the mechanism to undergo the antigen-antibody interaction. And these antibodies are being secreted by B cells of the adaptive immune system, mostly by the differentiated B cells. And these antibodies may occur in two physical forms. You know what are the forms? It is a soluble form that is being secreted from the cell to be free in the plasma. They can just wander in the blood plasma. And another type is a membrane bound form that is being attached to the surface of the B cell. So you can see on the surface of the B cell. and it is referred to as BCR, that is B cell receptor. And these BCRs, they are found only in the surface of the B cells. It actually does the facilitating activity of these cells, and thereby it will help to differentiate into whatever antibody factories, either as a plasma cell or sometimes it may act as a memory B cell. So, speaking about the antibodies, the antibodies are very much important for production of immunological response. To discuss about the antigen and antibody interaction that happens within our body. Yes, the antigen antibody interaction, which can also be called as antigen antibody reaction. It's a specific chemical interaction between antibodies that are being produced by the B cells or the WBC cells and the antigen during the immune reaction. The antigen and the antibody, they combine, for the process called as agglutination. So it is the fundamental reaction in the body by which the body is being protected from the complex foreign substances, namely the pathogens and their chemical toxins. Example, in the blood, the antigens are specific enough to win high affinity bond that are being binded with the antibodies to form an antigen antibody complex. And such immune complexes, they start to travel and transport to the cellular system where it is either destroyed or being deactivated. And the first description of this antigen antibody reaction was given by the scientist Richard Goldberg in the year 1952. There are several types of antigen and antibody, and each antibody is capable of binding to only certain type of specific antigen. And this specificity is mainly because of the specific chemical constitution of the antibody that is being present. And the antigenic determinant, which is otherwise called as the epitope, is being recognized by the paratope of the antibody that are present in the variable region of polypeptide chain. And the common type of antigen antibody reactions are hemagglutination, sensitization, hemolysis, neutralization, and precipitation. With this, we end this video and meet you again with another video pertaining to immunology.